Can you repeat that?
Okay, let's start over. Okay, so this is the 100. This is the male, and this is the female.
Yeah.
All the females were red-eyed, who's little me.
Okay.
The one male, because there's not a lot of males.
Yeah.
Is bronze. So, of course, when you have all dominant, all recessive, you get heterozygous. So
Yeah.
This is F1, so all of them have bronze eyes.
Dominant phase, right?
Yeah. So all the F1 offspring have bronze eyes, and yeah. Okay.
But then, let's say they practice incest, because they did.
Yeah.
2 heterozygotes.
Oh, I think I see where we're
Cross.
going with this.
So, this is what they're showing in cross 1. They're not showing the first cross. They're not showing this hole.
Yeah.
There's
Wait, that first one upper left is 2 big B's.
Yeah, I realized that. I was like,
Landy,
this
you're
doesn't
blocking
know.
it. Okay, thanks. Sorry, it's just your paper.
Okay, so this is F2. So you see
Sorry.
how there's some dominant.
Yeah.
So you see how some had bronze, some had. We didn't do the whole X, X, Y, Y thing.
Yeah.
So then they said true breeding normal winged males were crossed with true breeding stunted winged males. So this is the other gene. So it's
Okay.
like how we have the AA.
So when you
BB
do
thing, this is
probability,
This is the eyes, and this will be the wings.
Oh, okay.
This is what they're basically showing us.
Oh, okay.
Basically the same thing, but with wings.
Yeah, I didn't realize you were doing Punnett squares and not chi-squares. I was really
Yeah, but it
confused.
says
Well,
for C.
Yeah, for C you got to do chi-squares, which
Yeah,
so I was
but in order to do the chi-square, you need to know what the
What the
expected
genes are.
results are. So
Okay.
to get the expected, you would have to do this.
Okay.
I mean, I don't know if the numbers are totally correct. There was one male, and we have to do the whole X length, but this is the short, quick version of it.
So for
then-
1 and 2, it's basically you talk about the Punnett squares, so you provide those as diagrams and then
Yes,
analyze
you
them.
would
For A
say,
and B.
why are most males
A
a
and
certain
B.
way, or why are these people?
And cross 3 is the wing one.
Yeah, no,
Okay.
cross 2 is the wing one.
Oh.
So then cross 3.
Oh,
is
and cross 3 is a mix of both of those chi-squares.
Yeah, so it's like
or
how she
Punnett
says,
squares, sorry.
when she says, what would happen if we got one kid with AA? Like, what is the percentage? What is the probability we get both of these, right?
Mhm.
Well, then you would do 2 separate. So you would do this one, and then you would do the A, like that, basically.
Yeah.
And you would figure out, and this is how we got it. So you know how you've seen the P1 on the PowerPoint.
The P1 on the PowerPoint.
The P cross for when we were doing.
Oh,
the-
yeah, for
And
P's.
how it was like 9, 3,
I thought you were talking about like
3,
prophase 1.
1.
Yeah,
So
yeah.
This is basically what you're getting right here.
Okay.
Yeah.
So that's your 9331 data.
Yes.
So let's
So-
stunted wings be
huh?
dominant over normal.
Well, okay, here's it. True. All F1 had stunted. So so stunted. If all
Oh,
one
yeah.
had.
Stunted would be
If,
dominant.
yes. So for wings, we're gonna do W. Okay.
Mm-hmm.
So this is how we got F1. So these are the 100 females and 100. This is how we got F1. So this is a male. A male had stunted wings. This is little w. So little
No, wait.
w.
No, the man had normal wings.
Wait.
Oh, sorry. I'm sorry. Sorry.
No problemo.
So W. This is big W then. So big W equals stunted wings. Little w equals normal, which is weird. I hate when something like that. So the male had normal. The woman had big. So of course, you're gonna get all heterozygous. So this shows
No
that
homo.
all of them had stunted.
Yeah.
Okay.
No homo.
So then F2. Again, we did incest. So while they were doing incest for this, they were also doing incest for that. So
Naughty.
big W, big
Send
W,
that to me,
big
the
W.
video.
Yeah, I'm gonna send I'm gonna put the video on Skype and then you can email it to JJ.
Alright, little W. So then this is basically what we did to get this cross number 3.
Yeah.
So I'm gonna take, what are we comparing? What is this though? This is F2. So this is F2. So we're using these to get
Yeah.
the whole table. So what happens when we get dominant for the first dominant? So we're gonna get dominant and dominant for both.
Mhm.
So first, you're gonna get 3 out of 4,
Yeah.
and then you're gonna get,
Hold
and
up.
then
It says true breeding though. Would that mean homozygous dominant?
I think when you meant like true breeding would mean or homozygous recessive, like this is it.
Yeah. So if they're true breeding,
That's for the
bronchi-
parents though. That's not for F2.
Oh.
F2 is all over the place because they were
Okay.
technically mixed.
Okay.
So you're getting dominant for both. So dominant, and then you have 1, 2, 3
Mhm.
times
Yeah, no
3,
problem.
4 would equal 9, 16. So
Yeah.
that is the bronze eyes and stunted wings. That's the very first
Okay.
row. And then
No problem.
we're gonna get dominant again, 1, 2, 3. But we're gonna get recessive for the next one. So now there's only 1 right there.
Yeah.
So 1, 4. That is how you get 3, 16. So this is where I'm getting the 9, 3, 3, 1 from.
Yeah. 9331.
So then the next one we're gonna get red eyes, which is again recessive. So we only have 1.
Mhm.
And then we're gonna get dominant for the next one. So 1, 2, 3.
3. Okay. So it's also 3 out of 16.
So yes, this is also 3 out of 16. So the heterozygous, of course, is 3 out of 16. They have
Yeah.
this one.
And since the last one is
Recessive for
homozygous
both.
recessive for both, it would be
Homozygous-
1/16.
Yes. So it's
1/4
4 times
times 1/4 is
1/4.
1/16.
I hate that when I do that. And then 1/16. And then this is how you do the this is how you do the ratio. I forgot
Yeah.
what kind of ratio it's called. It's like the phenotype.
Oh, and then so these are the expected results, and then
These
for,
are the
they
expected.
give you the phenotype, the actual phenotypes for this
Yes,
one.
so she gave us the actual phenotype.
And
we
then-
came up with our own genotypes,
Yeah.
but it basically makes sense.
So we do
because-
a chi-square to compare the phenotypes with the genotypes that were expected.
Well, you
and
don't
see if
have
it's
to make a
accurate,
chi-square.
or-
I mean,
I think she said to make a chi-square though to.
Yeah, so I guess make a chi-square. You can copy it from the lab. So for the chi-square, you would have to do observed minus expected squared divided by
Yeah.
expected. So if you're doing, let's say, dominant for both, observed would be
So we have to compare everything. Like we compare like all the phenotypes with the genotypes,
yes. So you
and
would-
we see if the Punnett squares were accurate.
Yes, that's
Okay.
what you're basically doing. So you would subtract like the first row from like you would actually have to do the math because there are 100 females and 1 male. So you would have to
Yeah.
do the math. So in this, besides, so you know how we figure out the pedigrees, how they're like, male here,
Yeah.
here. I'm just gonna give them one girl, and then the dad has it, and then this person has it. So you would find out, first you would find if it's autosomal, so of course you would think, oh, well, it has to be recessive because if only one has it, then it has
Yeah.
to be recessive.
So we have to make a pedigree too.
No, I'm just showing you how,
Yeah.
how I came up with it. This is the AA. Of course, she would be dominant blank, but then you would have to know that then these would be AA. So that's basically how we did this, right?
Yeah.
But then you're like, oh, well, is it sex linked? So then, of course, you would get rid of this. We'll write over it. Then you write X, Y, X, 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 Y, X, Y, X, X. You'd say, oh, well, this is recessive, and then she has a big A, so she
So
has
we
big
did
A,
a little A.
big A. Well, yeah, I think it would be little A, sorry, because she, of course, gave it to her.
Yeah.
So then that's what you would also figure out because you have to figure out the percentage. And then whatever number you get from here, so whatever number you get from the expected, so it would be expected minus observed, so this table, so whatever table was across 3, divided by expected, so whatever you got from here, and I think this is squared, and that's how you do the first part. And
Yeah.
look at that chi-square lab. 'Cause
So for the part C, You're talking about how and why chi-squares. So you talk about chi-squares and how they compare
Yeah,
observed
so
results to
basically-
expected results.
Yeah, so
And
basically
how
what
it
we
would
did- yeah.
and how it would help is it would prove the Punnett square theory.
Yeah, so you would basically how it would prove the expected. Because you're
Yeah,
gonna do
yeah. It would
basically-
prove the expected because you would show that the phenotypes or the genotypes that you predicted in the Punnett scores were correct.
Yes, so basically you're counting the percent. So like how we did the M&Ms.
Yeah, with the M&Ms,
Remember,
like
in our
we
class-
were saying how far off the observed were from
Yeah.
the expected and how it actually wasn't doing a good job.
Like
The machine
one
was
of our
drunk.
classmate Amber, she had the pretzel.
Oh,
You weren't
yeah.
supposed to have any yellow, but
She
she
wanted
had a
yellow.
yellow. So
Hmm.
that of course was way off. So
Yeah.
basically that's what you're doing. Like seriously, what are those? So see, like look at that chi-square. I mean, I would go in for help. And then for D, sorry. That's but basically back to evolution. And if I discuss two factors of effect, whether the island slide population.
So, wait.
Okay, guys, I'm closing this because my thing is gonna die.
Yeah.
So you would, yeah. So
Right.
like, what you can do for this one is.
For D, so you would talk about so if the fly population, if it's close to other flies, then there will be genetic
So
drift and
basically,
blah,
the
blah, blah.
chi-square also goes to Hardy-Weinberg. It's basically like doing a Hardy-Weinberg. You're basically trying to prove that you know the population is not perfect. They're supposed
Yeah.
to be genetic and mutation. So that's basically what you're showing. If there is difference, it's not close to the perfect population, and you know there's mutation, then that's how you figure it out. And basically with the Hardy-Weinberg.
Because if your chi-square is far off, then obviously there's been a mutation or some
Yes.
kind of genetic
So
drift
that's
going
what they're
on.
saying in Hardy-Weinberg, what factors like genetic drift, like the bottleneck or whatever. So you have to identify two factors. So, what would be a factor, I guess?
A factor would be other flies in the vicinity, like if
Yeah, I guess like what you
if like the F2 cross has been contaminated, like if a different father fly came and
Yeah, or like he said genetic drift,
Yeah.
environmental factors, you know.
mutations.
There were 5 points of
Yeah.
Yeah. Okay. So.
Not an infinite population size.
Oh, okay. So.
That'd be a big one.
Look at 5.
Hey guys, can we
Yeah.
like kind of move on from this because we have like
Yeah.
5 minutes. It's 1:30, and if you guys want to study for the exam, I
Yeah.
can't really stay that much afterwards.
Yeah.
Kind of thing, so.
Yeah, no problem.
No, that's
So-
selection.
Okay. Yeah.
Look
Thank
at 5
you.
points for Hardy-Weinberg.
Thank you so much, JJ.
No genetic drift, no chain flow, no mutation, random mating, and no natural selection.
All right, there we go.
Yeah.
So
those are the 5 points.
so those are the 5 points. I'm going to cut off the video here. Thank you guys so much. I will send this to everyone right away. Yay!